Greetings, friends, fellow Earthlings, and explorers of the briny deep, and welcome to Ask An Astrobiologist, the show that celebrates the science and the scientists involved in our quest to understand the nature of life. I'm your host, Dr. Graham Lau, aka the Cosmobiologist, and we're brought to you by the NASA Astrobiology program and SegaNet.org. Now, before we start our show today, as always, a huge shout out to everyone out there, one who's watching our show live, to those who watch our recorded episodes later, and to those of you who share about our guests on our show, who talk with our guests on our show online, and who share on social media about the upcoming episodes we have. We really appreciate all that you do. A huge shout out this month to Antonio Molina at Athneos on Twitter/X, who shared about our show in Espanol for those who want to watch today and find out more about this incredible episode, which honestly, today's episode is going to be an absolute banger of a show. With the help of the Schmidt Ocean Institute, or SOI, we're about to embark on our first ship-to-shore episode of the show. Now, sadly for me, I'm the shore part of it, but we have our guests are joining us from the ship, from the research vessel Falkor Too, which is currently exploring the depths of the Atacama Trench off the coast of Chile. Leading the science of this mission are my personal friends and colleagues from the Blue Marble Space Institute of Science, Drs. Armando Azua-Bustos and Lauren Seyler. Dr. Azua-Bustos is also a researcher in the Planetary Inhabitability Department at the Centro de Astrobiología in Spain. He's an expert on xerophiles, organisms that thrive in dry places, and he's also spent a good deal of his research career exploring the Atacama Desert on land. Dr. Lauren Seyler is an assistant professor at Stockton University. She's an expert in the world of microbial activity, metabolisms in unique environments, and biogeochemical cycling. They're also just two of my favorite people on the planet. So I am so excited to now welcome them to our show live from the Falkor Too. Armando, Lauren, thank you so much for joining us.
Hello.
Hello, Graham. Thank you for having us.
I mean, it's super exciting. This is our first time doing a ship-to-shore episode, and so we might have a little bit of audio issues and video issues, but that's okay because this is incredible to see both of you out there right now. It looks like a beautiful day out there in the Pacific right now.
It's gorgeous. It's nice and sunny. The weather's been beautiful, and it is the calmest day at sea I think I've ever had as an oceanographer.
I love it. Now, I'll start off, Armando, this mission that the two of you are on and leading with many of your colleagues is about looking for living fossils of the Atacama Trench. Can you explain this idea, this concept of living fossils for our audience?
Well, actually, this started like a decade ago, in which I collected a number of evidences of microorganisms that have been finding in the coastal range of the Atacama Desert, some of them that look to be very ancient. So that led me to the hypothesis to say, well, what else is really ancient, in general terms, for all life forms that have been found in this area? To my surprise, a number of what we call living fossils, that is, species that were known before in the fossil records, that just by chance someone discovered them later, like the famous Coelacanthus, right? But in this case, a number of, I call them things with tails, like bivalves, like brachiopods, and other ancient organisms like crinoids, in which we know that are still around here and there in the planet. But in this case, it caught my attention there were so many findings just by chance, with people, typically fishermen or other people doing some other kind of research, finding them just in front of it. That in turn, when I also reported that all the microbial life that colonized Atacama, that, as you know, is also well-known Mars analog model, when trying to understand that phenomenon, how life colonized this really dry environment, I knew that whatever was finding inland at some point must have come from the sea. So that brought me to the question, well, what is going on there on the sea that would tell me that if the Atacama, not only the driest desert on Earth, but also the oldest, being 150 million years old, because the whole South American plate has been more or less in the same latitude, then it followed that the submerged part of the plate that goes into the ocean should be as old as well. So if people were finding these amazing species, say, hey, we have seen this in the fossil record, well, how come we are seeing it here crawling in the sea bottom? Maybe there were just those representatives just by chance was people finding for an entire ecosystem that should be that old. So I started getting people expert in everything that I did not have experience, like oceanography, marine biology, etc., 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 in order to bring in this amazing team of scientists, each one expert in each area, from geology to physics, etc., etc., in order to bring them there and say, let's see if actually this is true. We may find entirely ancient ecosystems. So for us, it's like a trip to the past. Right, 150 billion years old, how would the bottom of the ocean would look like? And that's what brought us here.
That's fantastic. I'm so glad you mentioned bringing together a diverse team with different backgrounds. You know, in astrobiology, especially through the NASA astrobiology program and the missions that we fund to go out and explore in the cosmos and here on Earth, it does require a lot of different disciplines. And so I guess maybe a fun question for both of you is what is your experience? Is this your first time, second time, 100th time out at sea?
This is my third time at sea, although it's my first time co-leading a cruise. But my first two cruises were in the Atlantic, in the North Atlantic, and we were primarily looking at activity in the water column. So this is my first time in the Pacific and my first time doing ROV operations. So I'm super excited to be involved in this project.
And in my case, it's even more incredible because this is my first time ever on any type of boat, except one time I rowed in a little lagoon in the middle of Madrid. That was it. So for me, it was an entire new experience from A to Z.
Wow, it is, Armando, it is a huge step from a rowboat up to this incredible research vessel you're on right now with the Falkor Too. For our audience who are watching right now, and for those watching the video later, we are going to have a live tour of the ship here in just a moment after I ask Lauren and Armando some more questions about the incredible science that they're doing on this mission as well. And so, Lauren, for you, I love this concept of looking for metabolisms in the dark, as you've called them, on this research mission. Obviously, you know, a lot of our audience should know that a lot of the primary productivity on Earth comes from sunlight and, you know, from photosynthesis. But now you're looking at things on the seafloor where it's extremely dark, a different pressure, an alien environment, really. What kinds of things are you specifically looking for on the seafloor?
Well, the thing that really interests me about the seafloor is, like you said, it is a dark environment. It's cold. It's under a lot of pressure. It couldn't be more different than the surface of the Earth and life as we tend to think about it. And what really fascinates me about this environment is that because there is no sunlight, there's really two main sources of food and energy for the organisms that live here. One is primary productivity from the surface ocean. So most of the productivity in the ocean occurs in this top 200 meters of the ocean where light can penetrate, and phytoplankton are doing photosynthesis and producing all this organic carbon. And all of that productivity eventually rains down to the bottom as something called marine snow. And we have seen a ton of that here because this is an incredibly productive area. This is a huge upwelling zone, meaning that water from the bottom of the ocean, which is very cold and nutrient rich, is seasonally carried up to the surface and fuels all of this primary productivity, producing all of this organic carbon, which eventually rains down to the bottom as these clumps of fluffy material that literally look like a snowfall. And so that's one major source of energy and food for organisms on the bottom of the ocean. The other one, and the one that fascinates me the most, is chemical energy. And that's the one that's the most astrobiologically relevant, because if we find life on other worlds in our solar system, chances are it will be fueled by the energy of chemistry, geochemistry. So the reaction of water and rock, fueling hydrothermal activity, fueling things like methane seeps. And so chemical energy fueling life is just so fascinating to me because it's so alien from what we usually think of when we think of the food web, right? We think about sunlight providing energy to plants so they can produce food, and then animals eating those plants and other animals eating those animals. But at the bottom of the ocean, there are entire ecosystems that are supported by the energy of chemistry, which is just incredibly fascinating. And if we find life on other worlds, we may find ecosystems just like those. Oh, it's incredible. It's the culmination of months of work and hours of work on the ship, but months of work of preparation. So a methane seep, for folks at home, is an area where, just like here, we have a subduction zone where you have one tectonic plate sliding underneath of another one, and it causes the plate on top to buckle and form all of these ridges and what in geology are called anticlines, places where the rock has been folded upwards. And underneath of these folds, methane gas gets trapped and eventually seeps up to the surface. And this is one really important source of chemical energy to the sea floor. Methane seeps can also form as a result of decomposition. So we were also prepared to find areas where methane generated by the decomposition of all of this organic material raining from above could also create one of these methane seeps. But we were really, really interested in finding the kind that are geologically formed. And so we were spending every night mapping the sea floor, gathering bathymetry data from the ship's incredible sonar system, looking at backscatter, which shows how much of that sonar penetrates into the sea floor and how much of it bounces back off of rocky surfaces towards the ship, and trying to find places where we might find a methane seep. We were looking for bubbles in the sonar data as well, but bubbles from methane seeps are generally a lot easier to find in shallower waters than this. So just because we didn't see bubbles, we still thought, well, they could still be down there. Maybe they're not bubbling very actively. Maybe they're too deep for us to see. But we found these places that looked really, really likely, and just went down with the ROV and spent hours crawling along the seafloor looking for signs of seeps. And when we finally found one, it was an incredibly exciting moment.
That's so fantastic. Now, for both of you, so this is Ocean Worlds Month through NASA. It's a time to explore not just our ocean world, the Earth, but also the many other ocean worlds of our own solar system, places where oceans have been detected, could possibly be, places like Europa and Enceladus, potentially Titan, Triton, maybe even Pluto, who knows? There's a bunch of possibilities for underground oceans and some of these other worlds out there. I wonder for both of you if you could speak to the importance of the research you're doing right now, not only in understanding life in other ocean trenches around the Earth, but also what is your vision for how this impacts our future research on other ocean worlds as well?
Well, that actually is a very interesting question because it's so highly related to what just Lauren said. You think about the icy moons like Enceladus, Europa, or others that we are discovering, in which we think we have this 6 layer of ice water, and it sounds redundant to say ice water, but at those temperatures you can have ice of many other things as well. You have the 6 layer of ice covering what we think there is internal oceans there, and at least in the case I think it's Europa, it even has more water than our planet. It has a lot of water. So given that you already know that it has ice water, then you have water there. That means that you have a source of energy that must be keeping that water liquid. And then typically what we know on Earth are the typical sources that we keep that liquid is the nuclei that is producing some kind of heat that interacts with the magma, that interacts with the water, while then you have this black smoke or the hydrothermal vents that produce a lot of water coming in contact with a number of minerals. Those minerals are able to be reduced or oxidized by different types of organisms, and then you have the primary producer, and you have the whole community that can suffer from them. So in the case of the Enceladus or Europa, for example, they will know, again, we have the 6 layer of ice. They are quite far from the sun, so you have little sun, so you would expect that if you have life and all that requires, water and all required by life in those systems, it may be the case that we know that we don't know nothing yet on the ecosystem that may be living in those moons there. And that it has been a huge discovery for science, because if you have proposed that maybe 20 years later, you would be called crazy. How come on, you can find life in the icy moons of Saturn? But now, actually, I think more astrobiologists might say, if we were to bet money on where we would find life, any type of life, from probably microbials, we know, we don't say Mars, we say Europa, we say Enceladus, because we know now that we have liquid water, which is the main ingredient for all life on Earth. So who knows? That's how we're looking at these environments as well. And we were just talking with a colleague last night when we were seeing the rover zooming on the ocean floor and seeing many species that the marine biologists have never seen before. We have this huge, it looks like a deep sea cucumber. It looks like a huge trilobite.
Yeah, the big black one.
Yeah, the big black one, black. And when they saw it, they jumped on their seat, opened their mouths, and pointed it to them. They looked at each other. What is that? So it would be more or less the same way we in 20, 30 years when we would send finally missions that were going to go through those islands and actually explore the oceans of Enceladus Europa and trying to maybe detect, who knows, an entire, completely separate from life, a deep sea ecosystem.
Yeah, certainly exploring the bottom of the seafloor with an ROV is a lot like exploring the surface of another planet. We know so little about the seafloor. I think we know more about the surface of the moon than we know about the bottom of our own ocean. And flying the ROV and seeing all these incredible scenes for the first time, it really did feel like exploring another planet.
It is eerily beautiful.
Yes, truly, truly.
I love that so much. And, so this Saturday as well is also World Oceans Day across the planet, a time to celebrate our oceans. So kind of, just a little bit beyond the research that you're doing right now, this amazing research vessel you're on, how do you see us doing a better job as humans in being involved in understanding our oceans, caring for our oceans, supporting our oceans moving forward? If there was like one message you would want to give people for World Oceans Day about the world's global oceans, what would you say?
I would say that we rely more on our oceans than I think most of us realize. The oceans of our planet are really the lungs of our planet. They respire, they breathe for us, they produce oxygen and carbon dioxide and generate energy and maintain the temperature of the planet. The ocean has been absorbing much of the carbon dioxide that we've been producing as a result of industrialization. And pretty soon the ocean is going to reach a tipping point where it just can't buffer that production anymore. And it's already starting to affect life in the ocean through acidification. It's really incredible because there were moments when we were on the bottom with the ROV looking at places that no human being had ever seen before, thousands of meters deep, and we were finding trash. And it was heartbreaking to see it, actually, because these incredible pristine environments where there's so much diversity of life, and it's so alien. I mean, imagine being a creature on the bottom of the ocean and seeing this huge robot come at you with all these lights on it. I mean, it must feel like an alien abduction, right? But there's still the signs of human life everywhere, even in places that we've never touched. And I think that sometimes we don't realize the impact that we have on these incredible, pristine places. And they were here long before we were, and they'll be there after we're gone. So we've been given a responsibility to be the stewards of this planet and to care for it as our mother, as our home, and we only get one planet Earth. So if we mess this one up, there's no other Earth for us out there. So the ocean is such a huge part of that. It covers most of the surface of the globe. It provides so many important processes that make life on Earth possible. There would be no life on Earth without our oceans. And so we have a great responsibility to care for them.
Oh, that's fantastic. I will say before we jump in, so we're going to do our tour here fairly soon. I have a few more of my own personal questions. As the host, I get to be a little selfish with things. One thing I do want to ask, Armando, you gave a TED Talk back in 2017 about your work on the Atacama Desert and your research in astrobiology. It's garnered well over 1 million views online now over these last 7 years. I wonder if you can speak to the importance of sharing your work through venues like TED and other experiences like that to let others know what you're doing in astrobiology.
Well, for me, it's so clear. What's the point of making and generating new knowledge if you don't share it openly at any time with everyone? It's as simple as that. What's the point of being a scientist if you stay in your own little world of the lab and so on? No, you have to share it.
Mm-hmm.
hey, let me show you this fantastic thing I have been finding around. And for me, it's like the whole deal, right? How you can, again, amass all those knowledge and just keep it to you for yourself. Come on, everyone.
Yeah, it's the best part of being a scientist is getting to share it.
I love that so much. Yeah, Carl Sagan said, when you're in love, you want to share it with the world, right? And many of us love what we do in the sciences. So I do want to share, we did a poll on YouTube for our audience before the show. We wondered like what people's favorite of the icy ocean worlds of our solar system beyond Earth might be. We gave them some choices: Enceladus, Europa, Titan. Maybe they could drop their own for Pluto or something else. Over 43% of the respondents said Europa is their favorite. Now, that said, one of our respondents left a comment too at scientist_ish, who is Ishan Madden, who is a graduate student who's reached out to me in the past to talk about astrobiology. Really wanted to push Titan as being a really likely place to explore for like a nitrogen-dominated atmosphere with stable surface liquids, complex organic chemistry at the surface. But it's also very different. It is a very cold world. It doesn't have the same kind of chemistry. The fluid on the surface there is not water. The fluid is methane and ethane and other hydrocarbons, which presents a very cool environment. Which then kind of brings me, Lauren, and your research, you've gone to a lot of really cool places to do research. You've been to serpentinization systems in Oman. You've been at sea. You've done some work studying these iron-rich bogs in New Jersey. I wonder if you can speak a bit to how exploring these diverse environments really plays into your own understanding of life on Earth and our search for life out there.
I think what I really enjoy the most about being a microbial ecologist and a biogeochemist is that it's constantly expanding my concept of what life is and what it can do. I think that we have yet to find the limit of what life really is. We have yet to really truly define it. And every time we think we have a handle on the limits of life, we find something that's even more extreme. And it's just fascinating to me. And it's the thing that I find the most fun to share with my students, that there are places where microbes can exist in boiling acid and these alkaline environments that are incredibly caustic at extreme pressures and freezing temperatures. I mean, it just fascinates me that life, to borrow a cliche, life finds a way, right? Yeah. And yeah, I think that's the most fun about my job, is that I get to go to all of these incredible places and examine what life is capable of doing in all of these incredible environments that are so alien, really, to our understanding as human beings of what is comfortable, what is tolerable, what is habitable. And that only expands our understanding of where we might find life elsewhere.
Fantastic. Now, this is kind of more for both of you. You know, those of us who maybe grew up watching Star Trek have always thought of space as the final frontier. But Lauren, as you mentioned yourself, you know, we've only explored the seafloor of our own planet to a small degree. I think roughly 20% of the bottom of the ocean has been mapped in high resolution, if I'm correct. But we still haven't really even explored much of the open ocean of our own planet. And so with a research vessel like Falkor Too, which is remarkable, and we're going to have a tour here very soon, and the ROV, I should say ROV means remotely operated vehicle for those watching and haven't heard that term before. For the ROV Sebastian, you know, we have these larger parts of our exploration of the ocean. But I wonder if you can help me tie this into space exploration a little bit. When you're out, like you are so separated right now, even though we're connecting over the internet because of how technology has progressed, you are still very far from the rest of civilization and humanity, very much like travelers going out into the cosmos to explore space. And then you're also using a robot to go explore a very alien environment. Does it feel like you're kind of also exploring space in a certain way while you're out there?
Oh, absolutely. Yeah. I mean, anytime you're at sea, you really are kind of in your own little microcosm, your own floating world in the middle of nowhere. And if you forgot something that you needed to do your science, well, there's no running out to the store and getting it. All of your needs are met right here. We're all living in this kind of mini laboratory / hotel in the middle of the ocean. Although as research vessels go, you can't get any more comfortable or high tech than Falkor Too. It is just an incredible vessel, and this has been probably my most relaxed time at sea in terms of just like having the comforts of home. But it really is still like being on a spaceship in a lot of ways. You know, you're isolated with a small group of people. We work every day, often around the clock. We keep watches. We have to time all of our work when we measure the water, when we put the ROV in the water, when it comes back up on deck. Everything has to be very organized, and everything has to be very planned out to the hour. And so it's in many ways a lot different than being out in the field when you're on land, because you really are kind of in this remote microcapsule, a scientific laboratory out in this vast expanse. And it's truly incredible. I mean, Armando, this is your first time on a research ship, so you could speak to this, I guess, the best, because this is your first time experiencing it.
Yeah, and actually, let's not forget that we have our room that is called Mission Control.
Yes, we do.
It's like watching a rover on Mars. But in this case, I actually was thinking about that because I was just watching how the rover was going down to the water there, and was seeing the water there, amazed how this little piece of machinery is going to go down, for example, tomorrow 4.5 kilometers down. And it's sending out like a probe. I was watching from afar, the same thing when you were talking with astronauts with the moon. So yes, it's quite amazing experience, quite different for what just Lauren said.
That's fantastic. So I'm going to do something a little bit different. We don't ever do this during Ask an Astrobiologist, but it's a ship to shore episode. This is our first of a kind for us. In the YouTube chat right now, Keith has dropped in and said, Eleanor says, "Hi, mommy." So I wonder if, since both of you are so far from your families, I wonder if either of you want to just share something for your friends and families back home.
Oh, hi, Eleanor. Hi, Josephina. Oh, hi, Keith. I think the hardest part about being at sea always for me has been being away from home. And this is the first time I've been to sea since I became a mom. And this is the longest that I've been away from my kids since they were born. So of all of the hardships of being at sea and all of the hard work that we've been putting into and the struggles that we faced throughout this entire experience, I think the hardest part has been being away from my babies. I love you both so much. And Keith, you are the most amazing husband in the world for supporting me as I explore the world and do all this incredible science. And you keep everything at home running smoothly, and you take care of our kids. And I could not have hoped for a more supportive partner than you. And I love you all so, so much.
Wonderful. I will say, Armando, real quick. So Rodrigo Azua has chatted as well, and wanted to say, please say hello to Armando from his family. They've been following the streamings with excitement. They're excited for you and the crew.
Thank you very much. I will say it in Spanish for them. Mi querida familia, ya verán que ustedes que cuando uno persigue los sueños se logran. Aquí estamos con toda esta maravillosa gente. Y por supuesto un gran abrazo y un gran beso a mi querida hija Margarita.
Oh, that's wonderful. Thank you so much for both of you for sharing that. And thank you to the families for tuning in and joining us here as well for Ask an Astrobiologist in our episode. We are going to make our segue now into the tour of the ship. Just before we do that, I want to give an acknowledgement real quick. One, to Schmidt Ocean Institute, who own and operate the research vessel we're on, the Falkor Too, and the Sebastian ROV. A huge shout out to Hannah Nolan and Sofia Pestenikova, who are helping us in the background right now. They're behind the camera, so our audience isn't seeing them right now, but there's some hand waves. There we go. Thank you so much for helping us out. For our audience who are watching, you can ask your general questions about the science and the research, life at sea, and all those things for Armando and Lauren, and we will do our best to get to those questions. I'm going to pepper our questions throughout the tour here, as well as towards the end. We'll have some Q&As for both of our researchers as well. But we're going to do a tour. And so as we go into each room of the ship here, then we kind of do our little tour around these different areas. If you have specific questions about the ROV, the hangar, mission control, the laboratories, any of that kind of stuff, the bridge that we're going to see, the research vessel itself, feel free to ask us, and I will do my best to get those questions over to our researchers, and we might even meet some other people along the way. So for our team on the ship, I think we're ready to go for our tour here then.
All right, let's go.
Okay, let's go.
So this is the aft deck. It's about the size of a basketball court. It is huge. There's Hannah. So you can see the crane that we have here that we can use to lift equipment onto the ship. It is massive, as you can see. And back here, we can store equipment. We have vans here for mobile laboratory space. And we have an arm on the back that we can use to lower equipment into the ocean. We're not using much of that right now on this expedition. We're mostly using the CTD and the ROV, which are kept in the hangar behind me. So let's go take a look at that space.
Oh, that is so cool. And I should say for our audience, some of our members of Ask An Astrobiology, our producer and director, Mike Toyon, and Sarah Treadwell, who is in the chat right now, have also been on research vessels in the past, which they've both told me that this is a much higher state-of-the-art research vessel, the Falkor Too.
Oh, this ship is absolutely
How does it compare for you,
incredible.
Lauren?
Yeah, I mean, I've been to sea twice before this, and the Falkor Too is just an amazing vessel. I mean, it is huge. It is so technologically advanced in terms of its capabilities, just anything you could possibly need. And in addition to that, SOI has an incredible science team on board that work with the scientists on every cruise that are just so knowledgeable, so helpful, so accommodating. I mean, they have just anything that we have needed, they have done their absolute best to make it possible. So not only do we have this incredible ship at our disposal with all of its capabilities, which we're so excited to show you, but we also have this team of very, very experienced people who have just done everything in their power to make this cruise a success. And so it's just been an experience unlike any other that I've ever had at sea.
Even the people that have many expedition like this one, when they first got into the ship, they were gaping mouths. Look at all the fantastic things we have around. Yeah, it is massive. It's beautiful.
Yeah. Even just as a place to live and work, it's just
amazing.
It's very clear that the folks at SOI, when they were building this new ship, they really took into account that not only are we working here every day, but we also have to live here every day. And so they've provided so many comfortable spaces for us, places to relax, places to sit together and watch movies. The food has been absolutely incredible, I must say. So this has just been all around a great experience. Totally first rate. So we're in the hangar right now. Over here is where the ROV Sebastian lives. And he's in the water right now. So you can see the winch and the cable that connects him to the ship. But when he's on deck, this is where he hangs out. And in a moment,
That's so cool.
Yeah, it's such a huge space. I mean, it's just awesome that we have this place that, the ROV can be. And whenever he comes back up on deck, we can get all of our samples off. And every time he comes back on deck, there's so much that needs to be done to turn around the ROV and prepare it for the next dive. And so we have lots of room to work around him and get what we need. Over here, we have the CTD rosette, or the CTD carousel. So this is a ring of Niskin bottles for capturing seawater from different depths. It also has a variety of sensors on it that tell us about the temperature, the depth, the pressure, the conductivity, the salinity and oxygen of the water that we can measure in real time as the CTD descends. And then as it rises back up in the water column, we can capture water from the depths that we choose and sample it. So this is a really important tool in oceanography that we've been using to study the seawater. And over here is
So real quick, Lauren, real quick for our audience, just to describe. So CTD is for. Could you describe what that means for our audience?
Conductivity, temperature, and depth.
Yeah, it's a tool that oceanographers have used for a long time, right?
Yeah. So you'll find one of these on pretty much every oceanographic expedition. Not all of them will use it. Some are more ROV driven, but we've been using both the CTD and the ROV on this expedition. So over here, we have one of these monitors that shows what Sebastian is doing right now on the seafloor. These are all over the ship, in the labs, in our rooms, in all of the spaces, so that if at any time the ROV is on a dive, we can see what the ROV is seeing in real time. And if we see something exciting, we can run down to mission control and say, hey, can you grab a sample of that for me? Or even just watch what the ROV is doing and enjoy this science from anywhere on the ship. So we don't have to be in mission control to watch the ROV. We can do it from anywhere using these monitors. Looks like right now they're picking up a rock sample using the scoop. There's a little shrimp.
Yeah, it's so cool. So we are actually cutting over to the ROV feed directly in our show right now so that the audience is seeing the arm up close and what they're doing.
Yeah.
I know we've had a joke already mentioned in the YouTube chat about the Falkor Too and the Sebastian as names. I'll allow our audience to share in the chat if they want to about where those names come from. We do have a question for our audience as well. So Sebastian is a very fun name. And so for our audience, we're going to drop a little poll in the chat just to ask everyone of, you see this amazing robot, but what would you name a robot for exploring the deep ocean if you had the opportunity to do so?
And maybe also ask, where do they think the Falkor and Sebastian came from, the names, huh?
Yeah. Who gets the reference?
I know at least one person has made a joke about it.
Yeah, so you can see on these monitors, not only can we watch the ROV, but we can also see the depth that the ROV is at, the temperature of the water, the salinity, and the oxygen concentration as well. And the cameras on the ROV are all in 4K, the side cam is at least, and so we can really see all of this in like really great definition. The resolution is great. The camera feed off of the Sebastian is just awesome. We've already captured so many beautiful images of the seafloor. Looks like the geologists are super interested in this rock right now. It's got some really cool banding, for sure.
Yeah, and not forget that they are the one you are seeing here is being moved by a person that will just show you mission control. I mean,
yes.
This person here moving that arm, that is taking place, the action 1.8 kilometers down us here. And
yes.
For me, again, it's amazing.
Yeah, there's actually 2 pilots that you'll meet when we go into mission control who control the ROV. One flies the ROV while the other one controls the arms. And it's all done through controls, almost like a video game. There's like a joystick that they use, and they can manipulate the arm of the ROV from mission control, and 2,000 meters down or more, it does everything that the pilots tell it to do. And you can imagine how tricky it is as an ROV pilot trying to manipulate this large robotic arm to pick up these often very delicate samples and place them on the ROV to bring them back up to the scientists. And sometimes we have to try multiple methods of sampling in order to successfully capture the sample that we want. But Sebastian's capabilities are just truly incredible. There's lots of space for different types of samples. We can take samples of rock, samples of sediment. We have bio boxes for taking animal specimens, rock boxes for taking rocks. There's more Niskin bottles on the ROV so that we can take water samples on the bottom. So every time the ROV comes up, he's just loaded with samples for all of our scientists to analyze and explore.
That's fantastic. I will say, so our audience is dropping lots of possible names right now, and I absolutely love it. I'll share all of those in a moment. I want to give them a chance to add some more. And for our audience who are watching, we're going to go into mission control here soon to see more of the rover operations. So please, if you have questions for Lauren and Armando, for those on board the Falkor Too about the rover, please start dropping those now. We'll do our best to get those over as well. It is so cool seeing this robot. Now, I've been told I think this is 1.8 kilometers right now in depth. What is the maximum depth your mission will be going to with the ROV?
Armando, that's your drive tomorrow.
The rover is able to go to up to 4.5 kilometers. And that's precisely the site we're going to go tomorrow to get as deep as the rover can get in order to see how the changing diversity and geology will look like when you go that deep. Just a half in the way to the deepest point of the Atacama Trench, which is about 8-point-something kilometers.
Mm-hmm.
And by the way, when we do that, the rover has to be especially prepared for the pressure that we'll have to endure under such conditions.
Mm-hmm.
So cool. Yeah, and we're getting back into a feed now from the robot. We're seeing it change out its tools here and drop its little basket. That's so cool to see. I think it is time for us to move on to our next site on the ship. But if the two of you are okay with it, as you're walking along, I'd love to ask some more questions from our audience as we go.
Absolutely. We're heading over to Mission Control next. Wet Lab next.
Okay, let's go to Wet Lab.
Awesome.
Let's go to the Wet Lab.
So just being cautious as you walk so you don't bump your heads or anything. Lauren, maybe this is a good question for you. Europa's oceans are gigantic. That's made that way because of tidal heating and flexing because of Jupiter. But how do we theorize having chemical energy available for life in an ocean like Europa? Do you have any ideas based on like microbial ecology and biogeochemical cycling, what might be possible there?
Well, I think that if there is life on Europa, it's very likely fueled by the process of serpentinization, which is a process by which water reacts with certain types of minerals found in ocean crust. And these reactions produce hydrogen gas, methane gas, and low molecular weight organic materials that can be used for life. And they are used for life on Earth at places like the Lost City hydrothermal vent on the Atlantis Massif, and also in ophiolites, which are pieces of oceanic crust that were lifted up off of the seafloor and placed on the continent. And I spent my first postdoc studying these processes and looking at the intersection between microbial life and the geochemistry of serpentinization. And so since we know that Europa has this very, very deep ocean and that there is likely a rocky seafloor at the bottom, if there's life on Europa, it may well be supported by the geochemical reactions between the water in the ocean and the rock of the seafloor that produces energy and heat and keeps the water fluid and could support microbial life there on Europa.
Fantastic. Now, are you taking us into Mission Control next?
We're going to go into the Wet Lab first, and I'd like to introduce you to some people in there. All right. So this is our Wet Lab. This is one of the smaller labs on board the ship, but this is where a lot of our water filtration takes place. And I want to introduce you to two of my new friends, Drew and Katie. Say hi. Say hi to the folks at home. Hello. Yeah, do you want to talk a little bit about what you're doing?
Sure. So I'm Drew Palmer. I'm a professor of biochemistry at the Florida Institute of Technology, and we're about 30 minutes south of Kennedy Space Center, so we get to see some awesome rocket launches. And
I'm Caitlin Hubrick. I just graduated with my bachelor's in astrobiology about less than a month ago, and I am starting my master's degree under Dr. Palmer, and I'm staying at Florida Tech.
And so part of our project is looking at microbiology, which is what we do in our lab, and we're also looking at biosignatures. So we're trying to look at some of the signatures of the life on the bottom of the ocean at these methane seeps and at ones that are may now be extinct. And so I think Lauren did a fantastic job of talking about that. And we're applying a couple of different methods. And we have one set up here, and I'm going to let Katie explain this process that we call solid phase extraction, which allows us to take our samples home in a lot smaller volumes.
Yes. So first we surface water off either the CTD or the ROV, and then I take the water samples back here. There's a few steps before we get to this. I first run the water through a 0.45 micron filter. We're hoping to trap our microbial friends that way. I then move the water to a 0.22 micron filter. We're hoping to trap giant viruses for one of our colleagues, Mara, in Spain. And after I'm done filtering through those two sizes, I get to the SPE, or the solid phase extraction. So first we take the water and we add acid to it to protonate it. And by doing that, what we're doing is we're making the water more hydrophobic in hopes that it will catch itself on the filter inside of our cartridge. And so each of these cartridges can take about a liter of water. What happens is I slowly run the water through here using the suction from a vacuum pump. And when it's done, we dry it and freeze it. And our plan is to take it back to our home lab in Florida. And we hope to find a bunch of chemicals. And our job is to figure out where did those chemicals come from, which one of our microbial friends is making those chemicals, and hopefully we can discover what metabolomic pathway are they using to create those chemicals. And that's kind of the job that we've been tasked with on the ship.
Yeah, so in addition to signatures of life, we're also interested in discovering metabolic pathways and products that are new to us. So new antimicrobials, new compounds that can be used for various purposes. So our lab has a lot of interest in sustainable space agriculture. So we actually work with bacteria that come from the International Space Station. And so we want to learn if we can use any of these microbes for the purposes of improving plant growth on Mars or the moon in future settlements. So that's been really interesting. We also got a great opportunity to go to the Atacama Desert before we got on the boat and have some samples from the desert to compare to some of the stuff that we're going to get here. So that's been a really great opportunity.
Very cool. Well, Drew and Katie, I would love to have you both on Ask an Astrobiologist in the future once you've done some of the work in your laboratory with these samples. So I will reach out after this show for more. But thank you so much for giving us a little breakdown of the science that you're doing in the lab as well. So cool.
Thanks, Drew. Thanks, Katie.
Bye.
All right, I'm going to take you guys into the main lab now. This is our largest laboratory space. Looks like a whole bunch of folks are working down here right now. So as you can see, we have a really, really spacious laboratory here on the Falkor Too at our disposal. It's equipped with fume hoods for working with dangerous chemicals. We have lots and lots of bench space here for all of our scientists to work on their samples. We have monitors, so we can continuously monitor the activities of the ROV as it's exploring the seafloor, and we get constant data feeds from that. Along this side, we have storage freezers at various temperatures. So we have ones at minus 80 degrees, which is where we store most biological samples that we intend to extract things like DNA, RNA, and proteins from. Over here, we have minus 30 degree freezers for other types of storage, things that don't necessarily need to be frozen at minus 80, like seawater samples, for example. And we also have a 4-degree refrigerator, which would be the same temperature as your fridge at home for cold storage. And I'd like to introduce you to a couple of folks who are working in here right now. My two new best friends, Antonio and Oscar. So Antonio and Oscar are geologists, and they have helped me so much throughout the course of this expedition, helping me go over bathymetry data and backscatter data and finding the best path for the ROV to look for methane seeps. So what are you guys up to right now? Can you introduce yourselves to the folks at home?
Hi, my name is Oscar Arcilla. I'm from the CAP, the Centro de Astrobiología in Madrid. I'm doing my PhD there, centered in the study of the Europa and Enceladus. And I'm here very grateful for being here in this oceanic expedition.
My name is Antonio Molina. I'm a planetary scientist also in the Centro de Astrobiología. And after working in few NASA missions in MSL, in Perseverance, also in InSight, being able to be here working with the Sebastian, it's a privilege to be able to compare the surface of other planets with the bottom of the ocean.
Thanks, guys. So have you found anything interesting down at the bottom of the sea that you want to?
Well, we are showing the magnificent outcrop of this site. So we are like, if we are watching our team in the Super Bowl. So for geologists, it's amazing to look at that kind of rocks at the bottom of the ocean, and that ancient rocks.
Yeah, we were saying the other day that the bottom of the sea is very homogeneous. So you are looking for these features that allows you to see what's below. So you are going to these cracks, these fractures, these reliefs. And now, yesterday, we were able to find these seeps that are something very interesting. It's like a small island of biodiversity and also geologic diversity. So in here, we are trying to find a similar feature in this new area. So fingers crossed, and let's see what's next. Yeah.
Oh, that's remarkable. Thank you both so much for sharing that with us.
All right, this is Guillermo. I'm very honored to be working with Guillermo. He's a local Chilean biologist and expert of marine invertebrates. And what are you working on right now? It looks like you've got a cool specimen.
Oh, well, I am working on this species of squat lobster that was very interesting because I have many species around the world. And some of them are proposals that they're indicator of methane seeps or another extreme environment. But now we can discover if this hypothesis is true or not.
Oh, wow. It looks like a little alien.
See, I have some point of view different about the alien people. When we see all this beautiful environment, many people say, wow, it's an alien world. This is the only world that we know how life.
Yeah, so true.
So please show me where what you are compared.
Oh, wow.
Very cool.
This is a world inside the other world.
Yeah.
This is very few knowledge. This is interesting. So many of the animal here was captured. Now I am working on this world too. That is animal very old because we have found this in the fossil registration. This is a pycnogonid. They're closely more related with spiders than crustacean, for example.
Mm. Wow, that looks like an alien.
Yeah, sorry, Guillermo.
Yeah,
does look like an
sorry,
alien.
Guillermo, it does.
Okay. Cool.
Very cool. Thank you so much. That's so incredible.
Yeah, thank you. See, we think that we may have found some new species on these dives. Yeah.
Yeah.
Tell us about that.
It's very difficult to say out of samples. So, that is a new species, but some of the samples we caught now are from animals distributed to Antarctica.
Really?
Very, very long from here. So we need analyze to determine if it's a new species or not. But just this causa make me think that it's a new species, because some amphipod and isopod and crinoid, or many animals that are not known here in this place. It's very, very strange.
Wow.
Are very alien from Antarctica.
Yeah.
Wow.
That's incredible. It's a good reminder, too, of how much we don't know of life on our planet yet either. There's so many things in the seafloor and in the water column.
Yeah. Thanks so much for showing us these. All right, where did we want to go to next? All right, let's head to mission control. All right.
Wonderful.
So
that sounds
go
fantastic.
Out this way. It's a little noisy in this hallway. So we're just gonna, gotta kind of watch your footing as you're walking along on a ship, even on a calm day. Hello. Why don't we, let's cut through this way, actually.
So I'll jump over to another question here. For Armando, Sanjoy Sam, a good friend of ours, wants to know if it's crazy to anticipate finding multicellular living fossils in this trench. Did others doubt you when you kind of first hypothesized that you might find these living fossils for this mission?
Yes. Well, because, this started by me as a kid looking fossils where I live in the Atacama, actually. And I was surprised that many years later I found reports on the actual living relative of such things that I knew as just stones. And we have 1, 2, only 3 reports in a huge area in front of the Atacama in the trench. So there was a good possibility of finding them but it's like going into New York City, trying to meet with a friend with no phone in your hand, right? And amazingly, and I think it was first or second dive, we found them all. Chitons, brachiopods, and other crinoids. I mean, there was even rocks that have the three of them all together. Mm-hmm. And again, it was, as I said, like traveling to the past as the Ordovicic ocean would look like. So that for me was a wow moment. Say, come on, I expected to find the things, but all three together in the same rock is a true bestiary of that time. So for me, it's yes, objective fulfilled.
Yeah.
Fantastic. So I understand you're taking us to mission control now?
We're moving through our multimedia room where we have a whole bunch of camera feeds.
Awesome.
And going into mission control, we do have to be a little quiet in here just so we don't disturb the pilots as they work. Yeah, because we are actively on a dive right now. So this is it. This is mission control. And first time I saw it, it reminded me of mission control at JPL, just smaller.
Mm-hmm. And to me, to the deck of the Enterprise.
Yes. So this is where our pilots work. So we always have two pilots flying the ROV, controlling the ROV, as well as a science technician. We have a partner right now is our watch lead. So she is directing the dive and also narrating the dive because we live stream every dive, every single one, no matter what time of day it is. You can tune in and watch what ROV Sebastian is doing whenever he is on the seafloor.
It's so cool. And I'll share that at the end with our audience as well, how to watch those feeds. I will say for our audience, remember, this is mission control, which sounds a lot just like space exploration. And so our feed is a little pixelated in the video, but we are slowly getting some better resolution here to see some of our awesome pilots and operators of the ROV. But, we are talking to a ship out in the middle of the ocean doing this incredible research with that robot down on the bottom of the seafloor. And so, so we're getting slightly better resolution here now. We can kind of see some of the monitors and the controls that they're working on. I wonder, could we actually see the controls the pilots are using? Could we get closer without getting in their way?
Yeah, absolutely. And we even have a pilot here, Zach, who has been incredibly helpful throughout this entire expedition.
Actually, I call him Magic Eye Sax because he had noticed things that many of us before.
Hello, everyone. I'm Zach Bright. I am co-piloting at the moment, but I am a senior supervisor here at Sebastian. And on the sticks here is Tyler, who is a very, very good, smooth pilot here, as you can see, cruising along. And you can take a look at his controls if you want and see all the little fine movements he's doing to keep everything nice and stable. It's a lot more work than it looks like, but it's obviously one of the funnest jobs you could possibly do, in my opinion. Certainly something I look forward to.
This looks super high tech.
Yeah, it's something I look forward to every day, of course. Sure.
Oh, yeah.
Now,
so these are dual T4 manipulators, which are hydraulically powered, state-of-the-art, very, very finesse-capable arms. We just picked up a king crab by delicately grabbing him in the backside with these 3D printed rubber grippers, and then picked him and put him into one of our collection boxes. Can you look down with the sit camera, and we'll show him? It's a very angry crab in there. But,
oh, gosh.
yeah, if you can. Yeah, let's look at him.
Oh, that's so cool.
So you might have to bring your camera over there, but he's right in the bottom left of that screen.
Oh, yeah, there he is.
You see him?
Wow, he's huge.
Oh, yeah.
Huge.
Oh, yeah. We had to sneak up on that fella. He was on the lookout for us.
I'll bet.
And then if you look over on this screen here, we'll show you the manipulators. So on this, each side, they're identical. They're titanium, 6,000 meter rated. You can, if you're delicate enough, you can pick up an egg with these. That's the kind of precision control they let you have. So they're a pretty amazing piece of kit. On this one over here, you'll see the tip of it is very different. And I'll show you here real quick. This is a custom-made one that we use for collecting delicate samples and corals and things like that. Can you turn on the lowers for me? Thank you, sir. So this guy here, you can see he has a blade, and that blade is for cutting the coral. And on the other side of the blade is two 3D printed rubbery pieces, and that allows you to still retain control of the piece that you just cut, because we try to take the minimum of any kind of animal sample we have to take, of course, because we don't want to harm anything we don't have to harm. I think each of these arms is in the neighborhood of 250 to 500 grand, if you were curious.
Wow. Yeah, it's incredible.
Every time I see the robot arms do anything, I think of 2001: A Space Odyssey and the remotely operate, or the little space bubble that Dave is in, moving the arms and stuff. Yeah, it's so cool.
It's so cool.
Thanks, Zach.
Yeah.
Yeah, thank you so much, Zach and Tyler. Thank you for letting us see the operations of the rover from mission control. That's so cool.
Yeah.
Absolutely. Thanks for watching.
All right, we can make our way back out of here.
For our audience at home, just a reminder, you can ask questions for Armando and for Lauren, again, about the ROV, the ship, about the science. We have a little bit of time left in our episode here. And so we are going to do another transition walk, I think, in a few moments here. Is the bridge our next stop?
One other person I wanted to introduce you to
go ahead.
quickly, and then we'll go up to the bridge.
Cool.
Over here we have Nick Skizas. He is our myofauna expert.
Hi.
Hello, how are you? Yeah, hello, how are you? My name is Nick Skizas. I'm a professor of invertebrate biology. I'm working at the University of Puerto Rico for the last 21 years. And I'm here in this expedition to provide my knowledge and expertise in benthic and also some pelagic invertebrates. Most of my work has to do with looking at biodiversity with different methods, morphological methods, genomic methods, and I'm also here because I have a lot of experience with previous marine expeditions. So usually my usual spot in the expedition is here in this place where I consult my colleagues about the possible ID of different species we see on the screen. And this is actually my favorite place too. Okay, this is where I like to be.
Yeah, every dive we were calling for Nick and Guillermo to come in here, like, we don't know what this is. Can you tell us? And they come running in.
Actually,
that's awesome.
You know, the movie.
So when I see those posts and it's like a Dumbo octopus and a crinoid and some of those things, we have some support from experts who are telling us what those things are when those posts are coming out.
Yeah.
Absolutely. Now, the majority of the work, of course, we're here, we're exploring. If we knew everything, we wouldn't be here, right? So a lot of the work, most of the work will be done back in the lab when we have the specimens and we're going to be able to work with them. So so this is just the exploration part and the collection part. Okay. But we do have, of course, a good idea of what what those group of animals are. But to go down to the species level, that's much more work. And okay. Yeah.
Thanks, Nick.
Yeah, incredible. Yeah, thank you so much, Nick.
Thank you.
Hi, Jess. Jess is data logging for me. Jess is my lead tech. She's incredible. She's done so much work on this expedition. And Muriel is our
awesome.
Our contact with the Chilean government. She's our observer on board. All right, and we're going to head up to the bridge, which is, I think, the other coolest place on this ship. All right. We're just going to start the ascent here.
And it sounds like you have a little bit of a walk. So while the two of you are walking, one, I want to share the ROV naming results because a lot of people who are watching right now live shared some possible names. And so as you're walking, I will share a few of those with you and the rest of the audience. Sanjoy Sam recommended MacGyver the robot. Ian Watson, who appears to have watched The Neverending Story, mentioned Artax. Rendering reality 3D animations, the user on YouTube said scratch and sniff. That's kind of a fun one. Like scratch the coral and take a sniff chemically. AC says Naughtybot. 4 dozen salamanders has said advanced Roomba. Peter Rassensberg guesses R2D2, Star Wars fan. We have a guest from Shun Mizuno who says Odin, the Ocean Deployed Exploration Network. The Scandinavian in me feels that one pretty well. Stephanie Haney says Angler Biter. Jacqueline Brick says ROV Subspace. Pete Woodworth says ROV Servo. I love that. I'm hoping that's an MST3K reference.
It absolutely is. I know Pete very well. That is totally an MST3K reference.
Awesome. It seems like we're all a community of nerds here and geeks because I'm loving all the references to other stuff. Awesome. And for those who are watching, we do have questions that we're going to ask here in a moment. If you have more questions, please just get those to us. Those who are moderating the chat on YouTube can bring the questions to our experts who are now on the bridge for us. Cool. And I think maybe I'll start with a couple of audience questions, and then I'll do our Faster Than Light segment. Do you want to introduce the crew here who are with us first?
Sure. So you've already met Armando and myself. Katie and Drew, who are our biosignature seekers. And we have Antonio and Oscar, who are geologists on board.
Awesome. And for those who are tuning in for the show, we're now speaking with our experts from the bridge of the Falkor Too. So a few audience questions first. The first comes from Arif Roni, who wants to know if the seafloor is being affected by climate change.
Oh, good question. I mean, I would say that pretty much every part of the world is being affected by climate change in one way or another. Certainly the deeper ocean, I think, is still buffered from those effects somewhat, but in shallower seas, we're already seeing the effects of climate change with things like coral bleaching, fish kills, you name it. And of course, the weather effects too are huge, and they've changed the way that ocean currents work, upwelling events, having these extreme El Nino, La Nina events. I mean, the effects are so far-reaching that it's hard to really sum it up in a few statements. But a lot of work is being done as we speak and has been done for many years to explore the effects of climate change on our oceans from surface to bottom, and to also project what those effects might be if things continue the way that they've been going. So yeah, it's a very serious issue that's on the mind of every oceanographer.
So I can add one thing that we have seen from working with marine biologists in my own department is that there are examples of changes in the ecological distribution of organisms where as the temperature changes on the surface, that changes the distribution of organisms that can move up and down the water column, and that can affect ecosystems really significantly.
Mm-hmm.
Yeah. And so reflection is we are just learning about this deep now. So it's a bit sad that we are not maybe even to analyze these deep waters before they are affected by all these changes that is happening in the planet.
In my case, it's surprising that we find entire hills made of carbonates down there. And what will be the changes that will happen to that process? The temperature of the sea start rising up.
Yes.
Who knows what's going to happen?
And as the pH of the ocean changes as well.
Very cool. So we have a couple of questions about how deep the ROV is going. Again, Armando, you mentioned that earlier, but we had someone who asked if you can reiterate how deep you are right now with the ROV and how deep it will be going. But we also have a question then on top of that from AC on YouTube. They want to know to what extent the biochemical pathways are altered by extreme pressures. Like, for instance, is there any modifying of protein enzyme folding structures and things like that in these very deep waters?
Well, do you want to start with the depth, and then I'll take the barrel file question?
Yes. Huh? Okay, so what?
Yeah. So how deep it?
Now, and this is very same moment 1.8, but tomorrow we're going to go 4.5, which is the limit of the rover that can go.
Kilometers. Yeah.
Kilometers. Yes.
Yeah, and certainly there are organisms that exist at the bottom of the ocean that are designed to withstand these pressures, and when they're brought up to the surface, they completely lose their structure. I think that the blobfish has become a pretty famous example. Everybody has seen pictures of the blobfish on the internet, and they're like, oh God, it's so ugly. It's all like droopy and sad looking. But that's because it's been brought up to atmospheric pressure when it lives at like 300 atmospheres of pressure, and so its body is just not designed to exist at pressures that low, and it just completely falls apart and becomes that blobby shape. And there are microbes too, of course, that are adapted to these pressures, and there are special culturing techniques that we can use to study these barophilic, high pressure loving microbes. That's basically these high pressure vessels that maintain microbial cultures under extreme pressure. And the last time that I looked into this, I attended a talk about barophyly. The report was that we have not yet found the upper limit of pressure that life can tolerate. They went as extreme as they possibly could go, and the microbes could still grow and thrive, which was amazing to me.
Oh.
Very cool. Let's see. We have a question coming in. If you're monitoring ocean bioacoustics at sampling depths, I guess, are you listening on the seafloor as well?
So we have that capability, but we aren't currently using it on the Falkor Too. However, Falkor does have the capability to use bioacoustics. So if we were interested in listening to animal communication, for example, at depth, Falkor does have those capabilities.
Very cool. So Muhoodles, hi Muhoodles, on YouTube wants to know how the discoveries and the research that you're doing is going to impact our exploration of worlds like Enceladus, and maybe even Europa for the upcoming Europa Clipper mission. So for all of you, how do you see your research impacting our search to understand worlds beyond the Earth?
Yeah, in our case, my research team is already making the first approach. Now we know we have found here on how, at what depth it will be the equivalent, considering that you have less gravity in moons like Enceladus and Europa, and the relation between pressure, gravity, and depth of the ocean, and what kind of adaptation you would look for. That's what kind of biosignature you may look for if you were to go to such worlds. So we're already making the first approach now that we know, okay, so at this depth we have found these guys, at this depth we have these other guys, and so on. Here we have carbonate, here we have other type of rock, etc., etc., etc. Here we have pressures of this type, temperature of that type, in order to make the first proposal of where would you look, where would you like to go in this ecosystem like this one, in order to say this is the most that it looks like in terms of environmental parameter to what you would expect to find, for example, in the moons of Europa and Enceladus.
And also the exploration of Europa and Enceladus will look very similar of we are doing here with the Sebastian. And also it's a proof that we can do it, but we need to research and to do a better project to send a mission to Europa or Enceladus. For me, that my focus is in the research of the icy moons. It's very important to be here to know who this kind of ocean is the behavior of the salt, the interaction between rocks and the salts, the existence of methane shift, that it could be one of the best focus to the astrobiology. That is the main work of Armando, Antonio, and I. So it's very excited to be here. We have many data that will be useful to the research of icy moons in, let's say, Europa, Enceladus, or wherever other in the solar system.
And actually, I forgot to mention that it was supposed also to be a instrument testing mission. We were trying to get this Raman spectrometer, an instrument that will detect in situ, down there, the mineralogy or whatever we're looking for, like our own geologists down there, right? We couldn't get it in time, but it will come. Why is that important? Because those more or less the same approach we want to then take to these icy moons. So if we are able to do that kind of research at the pressures with all the technology that means to do that, and then we can also dream about maybe building such an instrument in order to ship it somewhere else.
I think our work in some ways contributes to that, because what are the signatures that you're going to look for? And so by organics, we think universally will be signal molecules that we might be able to find for either active biology or at least the fingerprint of remnant biology.
Yeah. I started my career as an oceanographer and became an astrobiologist as a postdoc. And one thing that I noticed right away as I sort of transitioned into this new field is how similar they are in the fact that they're both incredibly interdisciplinary. Oceanography and astrobiology really go well together. It's like peanut butter and jelly, just a match made in heaven. I mean, oceanographers and astrobiologists think about global scales, geologic time scales, or we're thinking about things on a planetary scale that just really, really, really applies well to thinking about other worlds. And in particular, because the search for life is the search for water, so much of what we apply as oceanographers applies to the search for life on other worlds. And I think what's really interesting about this particular cruise is that normally, on a research expedition like this, you have a lot of oceanographers who are dabbling in astrobiology, and here we have a science crew that's primarily astrobiologists who we trained to be oceanographers in the space of about a week. So it's been an incredible experience.
That's so cool. And again, I love the interdisciplinary nature of your entire crew. You have a large group who are working together from very different backgrounds. Another question from one of our audience members who's watching is if you can explain some of the differences, maybe both in the biology and the geology of the ecosystems from high temperature and low temperature ocean environments. I think they're thinking primarily of things like hydrothermal vents versus where you are right now in these lower temperature methane seeps, serpentinizing systems, and things like that.
For me, basically, they're the same. As Lawrence mentioned in the beginning, is energy, is life getting energy from chemistry directly, right? Microorganisms that were able to learn how to use iron or sulfide, methane or whatever compound in order to reduce it, oxidize it or whatever, they have to get ATP, and then they be the primary producer. So we have these seemingly very different ecosystems, which are basically, for me, the same. The concept is the same. Of course, one is being made. You find one where the plates are being made. Here we're finding the opposite, where the plates are going on top of each other. But for me, it's basically they are the same. They have different temperature ranges, and that's it.
Yeah, I think you could certainly look at these two very different ecosystems and talk about how they differ chemically, how they differ in terms of temperature. But ultimately, the reason why they're both so interesting to astrobiologists is because they provide a source of geochemical energy for life. And in particular, hydrothermal systems, whether they're low temperature or high temperature, could be a great place for a life origin to happen. And so, yeah, I would agree that, like, ultimately what we're thinking about is geochemistry for life, and it really depends on what's available in terms of the planetary structure. Maybe you have a planet that doesn't have tectonic activity, and so hydrothermal vents are highly unlikely. But serpentinization, for example, where you have water-rock interactions, could still be possible. So you have these lower temperature hydrothermal systems as opposed to like black smokers. But in the end, like it's the geochemistry that fuels the energy for life and an origin of life.
And let me add a beautiful example that was discovered a few years ago, in which people found microbial life doing photosynthesis at the bottom of the sea, because these guys invented a type of chlorophyll. I don't remember what it was, bacterial chlorophyll or rhodopsin, that were able to transduce light being produced from the shining lava, infrared radiation, into ATP.
Yeah.
So that's it's an amazing example of how, again, as you said, life finds a way. So who knows what we're going to find elsewhere?
Yeah.
Even from a former place that was potentially hospitable to life. So when we think about Mars at one time we believe had a warmer core, right? And so how could that energy have supported life? So I think what we learned here is, immediately adaptable to our search for life beyond, for sure.
Yeah.
That's so cool. So I'm being told right now by folks from SOI and my producer that there is an octopus on the live feed from the rover right now. I wonder if it's possible for us just switch over and see it for a moment for our audience to see what the ROV is looking at on the ocean floor. I think our pilots now are controlling the ROV so we can have a look down. Hopefully we can still see it. They're going to try. But, you know, sometimes in ocean exploration, you see the things you see in a moment, and then they can also disappear right away. Yeah, I'm not quite seeing it right now. I think we might have missed it, unfortunately. That's totally okay, though. So we do have a few minutes left. I want to say there are more questions coming in. So for all of you on the cruise, I know that there's a website for the mission, and all of you, you know, have ways of sharing. I'd love for our audience, if it's okay, to like reach out, find ways to learn more. I will say for our audience that SOI has really great video feeds of all the dives. They have a lot of information on the website from SOI about this mission and other missions they have. I know some of our audience members had questions about things like what kind of connection they're using right now for having all of these wonderful experts join us on the internet right now from across the world. They also have questions about like the fuel for the Falkor Too and some of the things like average time of duration of cruises. But I think since we only have a few minutes left, I do want to finish up on what we call our faster than light segment now at the very end of the show. So these are our questions that we have for our experts to highlight a few of the ideas that come up in astrobiology all the time, the questions that people have about what it's like to be an astrobiologist. Many of these are my own personal questions too, because I think they're a lot of fun. And so I'll start off for Armando and Lauren. What is your favorite thing that life on Earth does?
Oh,
very easy. What I've said several times. What I have seen in the Atacama, at least, that you find the most surprising adaptations. Every time I go sampling in some new site, I find something really unexpected. Every time, the same thing. So the reports that I report as in science, they come very easily because it's so novel. I mean, you don't have to do much work as to properly describe it in order to find, hey, I just found the first member of entire aquatic microalgae that is able to live on the bottom of the world, stuck to a spider web, because they're on the spider web, they're able to trap water from the environment. So for me, it's how life adapts to the most amazing conditions, the most unexpected conditions.
I think for me, as a microbiologist, the thing that fascinates me most about life is the connection between life and the non-living world, and how sometimes those that delineation is kind of fuzzy, especially at the microscopic level. Microbes are constantly taking non-living matter and turning it into living matter, and then decomposing living matter back into inorganic material. And it's this constant cycling of minerals, water, gases into living material that just absolutely fascinates me. And I love that really everything on this planet, life and all of the non-living things as well, are interconnected. And I just think that that's incredibly fascinating.
Wonderful. You're out at sea right now, and you're seeing all these incredible organisms, from microorganisms, and you know, as we heard from Drew and Katie, collecting viruses and other materials, but also larger creatures. If you could communicate with any non-human organism on the Earth, what would it be and why?
This is a tough question. Off the top of my head, I would love to be able to communicate with an octopus. I know I'm a microbiologist, and I always stand microbiology, but octopi are just fascinating to me. They're so intelligent, and I would love to be able to have a conversation with one.
In my case, I think, let's just remember that we actually did that. A team of researchers that a week ago reported that were for first time speak whale to a whale. And they called the whale to come, and they have this conversation. Of course, they didn't understand much what the whale was saying, but we are in a pathway in order to have the first interspecies communication, which also brings us to the world of astrobiology, because if we haven't yet learned to communicate with a fox or a whale, how do we expect to find with an alien being if it suddenly lands in front of you? So that's, for me, again, it's a very interesting question. In my case, I would like to talk with crows. Crows, yes.
Crows?
Yeah.
Yeah.
They're very smart as well.
From my end, it looks like I just lost the feed just momentarily here. So I think we're going to keep working here on trying to figure out how to get the feedback. I've lost audio and video. So for our audience tuning in, these are fantastic questions. Thank you so much. I can't speak to the exact kind of connection they're using or the fuel the Falkor Too is using. For AC, earlier you asked a question about if slowly decompressed, can these life forms from the deep be brought back to the surface, and can they adapt? Of course, the answer to that is yes in some cases because we've seen them already on the ship. But in other cases, there are some instances where things have adapted so much to that deep sea environment that you can't bring them back to the surface, not without drastic changes to their metabolism and biology. Also, as Lauren mentioned earlier, the blobfish, which does change drastically when it's brought from the deep sea back to the surface. It looks very, unfortunately, ugly. It was once voted the world's ugliest organism, which is so unfair. That's like putting a human face into like a giant wind factory, wind turbine kind of thing, and like blowing your face back and being like, well, you look ugly because it's a different environment than you evolved to. I will say, so I think our researchers came back to us. We do want to share real quick. We put a poll in our YouTube chat just asking how fast you think the ROV Sebastian might maneuver underwater. We had answers like 3 knots, 5 knots, 1 knot, or 10 knots. Of the 28 who responded, you got it right. It is 3 knots, which is actually pretty darn fast for an ROV underwater. So Lauren and Armando, it is great to see you both back here again. We have just a couple of minutes left. I think I'll do 3 more questions if that's okay. So Lauren, this one is directly for you. Since I know, since you and I are both very nerdy, and we've talked before about some of our nerdy interests, I'm wondering if you can share with our audience what stories have inspired you to want to explore more about life in the universe.
Oh, wow. Oh, I love that question. Oh my goodness. So I teach an astrobiology course. It's a general education course that's stocked in. So it's available to everybody, not just science majors. And there are 3 movies that I show in that class, just because I think that they're great examinations of alien life, but also because they were so inspiring to me and so formative to me. And that is Ridley Scott's Alien, James Cameron's The Abyss, and Stanley Kubrick's 2001: A Space Odyssey. And in particular, those last 2, The Abyss, I think made me want to be an oceanographer, and 2001 made me think about space as a kid in ways that I never would have, I believe.
Very cool. I love those films as well. They're fantastic. Armando, my next question is for you. You've had a remarkable career already. You've done so much research in the Atacama. You've been a researcher in Spain for some time now, and also a member of Blue Marble Space Institute of Science, like Lauren and I. You've had a lot of experience in astrobiology and understanding our Earth. But I'm curious, as we look forward to the future of astrobiology and understanding things here on Earth and out there in space, what is something that excites you about the future?
Oh, I'm kind of sad about the future because I'm going to miss so many great missions. I'm going to visit Europa and Enceladus and the colonization of Mars. Probably I will be very old when we colonize Mars, and probably my grandkids will get to see how we step on the surface of Enceladus, for example. So for me, it's like, oh, come on, give me 100 years more to live in order to see all these fantastic places we're going to visit. But I think doing this way, along Lauren and all the amazing team of scientists, and along the crew of the Smithsonian Institute, the Falkor and the Sebastian pilot, etc., we're paving the way in order to do that. I mean, how we're going to think about exploring Enceladus if we don't have this kind of experience beforehand.
Absolutely. Yeah.
So I will be part of the story some way or the other.
Fantastic. So we are at the very end. If I could have just 2 more minutes real quick from both of you, what is an unbelievable science fact that still blows your mind?
Oh,
that's a good question.
That's a tough one. I'm going to say, because when I learned this as a first-year graduate student, it blew my mind, and it still blows my mind every time I think about it. There are more bacteria in the ocean than there are stars in the universe.
Love it.
Nice. And in my case, is how little we know and how different species interact. Now we know, for example, all this network of fungi that are in giving different trees of different species in a forest to communicate with each other, things that we didn't even dream of how life is talking to each other. We are not aware of what they are saying. For me, the next big field is on this interspecies communication. That thing that you didn't even dream that would be taking place, like as she mentioned in some part, Lauren, whether the microbiota, or my case, if I don't have the proper microorganism or the wrong microorganism, I might get a depression because it's interconnected and talking to my brain. So this field of knowledge for me is the one that this is quite amazing.
Awesome. Well, thank you both so much. Thank you to Hannah, to Sophia, who are in the background with the camera, who've been doing so much work in helping us to highlight all of the researchers on board the Falkor Too. Thank you to Drew and Katie and Oscar and everyone else who joined us. I'm so glad we had a chance for our audience to hear from so many voices of people who are working on Falkor Too to make your mission possible. Now, I will say at schmidtocean.org, the website for SOI, there's so much information about this mission, the living fossils of the Atacama Trench, as well as many other missions, the cruises that have happened. There is the live feed we've shared in the chat. You can find it on YouTube from Schmidt Ocean. To watch all of the live feeds from the ROV Sebastian when it's underwater. If you want to check out Lauren's Twitter/X, she's been sharing a lot of really cool stuff there. She's at Dark Microbio on Twitter or X. A lot more to learn there. It's been so great having both of you join me for Ask an Astrobiologist. Thank you so much.
Thanks, Graham. It was great chatting.
Bye-bye.
Awesome. And for all of our audience, as usual, we're so happy you joined us. We're going to share ways that you can keep up to track with upcoming episodes of Ask an Astrobiologist by joining the email newsletter from NASA Astrobiology. You get lots of information there, opportunities and events, and all of the things going on across the realm from the NASA Astrobiology program. Of course, you can always reach out to me online if you have questions for me or for our guests from the show. We are always happy to try to answer them for you. If you can't get a hold of people from our show, just reach out to me and I can help you to answer questions as much as possible. So for everyone tuning in for this, our very first Ship to Shore episode live from the Falkor Too with Drs. Armando Azua-Bustos and Lauren Seyler, thank you so much for joining us. And until next time, remember, stay curious.